Tonight, I'll be sharing some evidence against evolution. And if you disagree, you're welcome to join the stream and challenge me. I'm going to give a short presentation, and then anyone can join the stream and share your agreement, disagreement, your evidence for evolution. So I welcome you to click that link and join me. Now, there are so many problems with the evolutionary timeline, and I'm not going to discuss them all tonight, but I'll briefly mention there's problems with the molecular divergence dates that not matching up with the fossil dates. There's problems with mutation rates with animal species. There's problems and genetic diversity with animal species. There's problems with the Y chromosome in humans not matching up with the evolutionary timeline. But tonight, I'm going to be focused on the problem, the discrepancies in the evolutionary timeline versus our observations of mutations and the rate of mutations in mitochondrial DNA. So, mitochondrial DNA, this is something that you have in all your cells, and it's passed on by your mom. It's only passed on by your mom, and it's not part of your nuclear DNA. So it makes it actually pretty interesting to look at because it's not undergoing recombination and all the complications that come with the nuclear DNA. So mitochondrial DNA, about 40 years ago, we started being able to really look at it and see the number of mutations in humans across the world. And it turned out that there's not that many differences between all humans who ever lived. At max, it's like 120 differences in the mitochondrial DNA between all humans. And there's like approximately 16,000, little more than that, in human mitochondrial DNA. So that's not that many differences for all the time that humans have supposedly been diverging and populating throughout the earth. So this was really interesting. And so we noticed, and usually there's more like 30 to 80 differences, but max 120. And so the scientists said in the beginning, like, well, this must mean that mitochondrial DNA mutates very slowly. It has to be really slow because there's just not that much diversity. So they were like, how can we figure out how slowly it's mutating? And their idea was to calculate it based on the human chimp divergence. So they say that we diverged from chimps from 5 to 7 million years ago. And so they tried calculating mutation rates based on how many differences there are today, and then how long it would take to get those differences if we went back to that common ancestor of humans and chimpanzees. So what did they find? Well, they had their evolutionary rate that they came up with based on that human chimp divergence would give us about one mutation in human mitochondrial DNA for every 600 generations. That means your mitochondrial DNA is going to look exactly like your mom's, exactly like your grandma's, exactly like your great grandma, great grandma, great grandma, so on and so on. For 600 generations, it's not going to change at all. Okay, so that's what they predicted on their evolutionary timeline. But what happened is that when they actually started observing pedigrees in real time, where they would actually take samples from grandmas and moms and daughters and track it according to genealogical records, going back even further than that, they realized that actually the mutation rate doesn't match up with the evolutionary expectation. In fact, the studies that have been done have found rates that are more like 1 in 25 generations will have a mutation, or 1 in 40 generations. Somewhere between 25 and 40 is what most of the studies that have been done have shown, that actually we do see mutations happening a lot quicker than that. So That's a problem for the evolutionary timeline. And it also is something very strange. And I actually I want to just substantiate this with quotes from these papers because this phenomenon is real. This difference between phylogenetic, which is evolutionary, and pedigree rates. And so I just want to read some from these papers. These are like a good sampling of the papers that have been done on mitochondrial DNA mutation rate studies in the last 25 years. So this is going back to, I guess this one's actually from even 1996. So the simplest, this is from a paper in 1996. It says the simplest explanation for our results is that the phylogenetic approaches have substantially underestimated the rate of mitochondrial DNA divergence. Okay, we got a paper from 1997 saying 10 substitutions were observed, resulting in an empirical rate of 1 mutation for every 33 generations. This is roughly 20-fold higher than estimates derived from phylogenetic analyses. Going on to a paper in 1998, evolutionary studies led them to expect about 1 mutation in 600 generations, 1 every 12,000 years. So they were stunned to find 10 base pair changes, which get them a rate of 1 mutation every 40 generations. Going on to 2000, recently, this mutation rate has attracted considerable attention, in part because of differences in the published estimates, which span 2 orders of magnitude. Now, some people may be saying, hey, this is so long ago. This is like 30 years ago. Yeah, this problem continues to this day. So 30 years later, here we are. And I'm going to keep going with year 2001, combined with other familial studies. Our results add up to 18 substitutions in 1,729 transmissions, confirming earlier findings of much greater mutation rates in families than those based on phylogenetic comparisons. So again, the phylogenetic does not match up with our observations. So 2005, the observation of differences between various estimates of the mutation rate in human mitochondrial DNA has promoted an intense debate. Why has it promoted an intense debate? It didn't match up with the evolutionary expectations. 2009, a calibration that corrects for the evident time dependency of the mitochondrial DNA mutation rate is urgently needed in order to reestablish credibility of mitochondrial DNA in human evolutionary research. That was 2009. Now, all the way to 2024, this shows you this is still an issue going on today. Pedigree-based estimates are substantially higher than those inferred from phylogenies, both in humans and in other species. Tonight we're sticking with humans, but there is also things to be said about the problems with mutation rates in other species. So what's really interesting is that actually the mutation rates observed fall very conveniently on the timeline of the biblical genealogies. You know, if you just read the Bible and you look at the genealogies, they've got genealogy from Adam to Jesus Christ. And you've got, if you calculate out the number of years for many of the patriarchs, it has the number of years that they lived. And there are there's several different possibilities. Like there's the Septuagint, which has slightly different dates than the Masoretic. So it depends on which one you choose. But our date for when Eve would have lived, according to the genealogies, is going to be somewhere between 6,000 and 8,000 years ago. So it's very convenient that actually that's what the mitochondrial rates, if we just take them back, then they fall nicely onto this. And I'm going to read some of these papers here. This was in Science, and there's one from Nature, but it says this: regardless of the cause, evolutionists are most concerned about the effect of a faster mutation rate. For example, researchers have calculated that mitochondrial Eve, the woman whose mitochondrial DNA was ancestral to that of all living people, lived 100 to 2,000 years ago in Africa. Using the new clock, she would be a mere 6,000 years old. Now, of course, this goes on to say we don't accept that she lived 6,000 years old, but that's what the actual rates would show. So here's another study, and this was in Nature, published in Nature. Using our empirical rate to calibrate the mitochondrial DNA molecular clock would result in an age of the mitochondrial DNA most recent common ancestor of only 6,500 years ago, clearly incompatible with the known age of modern humans. Even acknowledging that the most recent common ancestor of mitochondrial DNA may be younger than the most recent common ancestor of modern humans, it remains implausible to explain the known geographic distribution of mitochondrial DNA sequence variation by human migration that occurred in the last 6,500 years. So he's saying, look, this does our mutation rates, if we follow the empirical rate, it gives us one woman who lived 6,500 years ago that all humans came from. And we know that doesn't match up with the evolutionary timeline. And it also doesn't match up with what they think about the geographic distribution of human population, because you've got people all over the world that were supposedly there 20,000, 30,000, 50,000 years ago in these different places. And yet they've all got to come from one lineage that, if we follow the empirical rate, was 6,500 years ago. Now, and then finally, and this is the Howell 1996 paper, it said, the present results, as well as those of Lundstrom, suggest that either a much more recent origin of modern humans, or that the evolution of the mitochondrial genome is not a reliable evolutionary clock. Okay, so they're saying, look, maybe we just can't use mitochondrial DNA as an evolutionary clock. Okay, so they're ready to throw out the empirical and take, the things based on assumptions of when these things split, if that's necessary, or there is a more recent origin of modern humans. So maybe that should be considered. At least that's what the author is saying. So now, I don't want to leave anybody thinking that there's no attempted resolution at this. Okay, so how are people who continue to believe in evolution, how do they reconcile? How do these scientists reconcile this discrepancy between their evolutionary assumed dates and the actual empirical observations? Okay, so here are some suggested reasons for the discrepancy, and I will try to go through these quickly and simply, but this is getting into the weeds. Okay, so they would say some of this high mutation rate could be because we could be actually counting somatic mutations instead of germline mutations, meaning when they take somebody's blood and they look at it, they might be seeing mutations that are just happening in the body, but won't be passed on to the offspring, because the offspring, that's coming from, the reproductive cells that are already created, and they might not have those mutations. So that might be what we're doing. We might be accidentally calculating into the fast rate somatic mutations. Another thing they say is that, mitochondrial DNA, and especially in the region that they like to test, because I kind of shortened the presentation, but actually what they found is that if you take the whole mitochondrial DNA and you have different parts of it that have different rates of mutation. And so you have some parts that seem to click, go the clock ticks slower, and some that go faster. And so they actually have kind of focused in on one region that they think is a better clock because its mutations are neutral. And what I mean by that is, so if you have like if you have coding region in your mitochondrial DNA, and let's say you get a mutation in that coding region, it might kill you and then it won't be passed on. So, you know, they realize that the part that makes a better clock is the part that's the non-coding region, where when a mutation happens there, it's likely not to kill or harm the person. And so the mutations can collect more substantially without causing a problem. And so they think of those sites as more neutral. Okay, well, the issue is that, you know, one of the things that they say may be causing the elevated mutation rate is that, there might have been so many mutations that in this region that they're testing, maybe some mutations actually went back to the original. Like maybe there's been so many mutations, it actually like turned over and came back to its original spot. So we can't tell that there was ever a mutation there. Okay, so that's the saturation idea. Okay, another issue that they say may be causing the discrepancy is heteroplasmy that will eventually be lost. And let me explain that. So when they take the mutation, when they take the mitochondrial DNA and they look at it, in some people, they don't have all the same mitochondrial DNA. Some of them have some mitochondrial DNA that is not, that's all like that has a mutation, and some of it that doesn't. So a particular mutation. And so it's not like you have many mitochondria in your cells. And so you may have some that is mutated in one area and some that's not. And so they say that, and they call that, when some are mutated and some don't have the mutation, that's heteroplasmy. And they say, if we just go by, this mutation rate, maybe we're not accounting for, heteroplasmy that will eventually be lost. Because just because you have some mitochondrial DNA with mutations doesn't mean that you're going to pass that on to the next generation. It might be lost in the next generation. Maybe your offspring will only have the non-mutated version. And so that could be what's causing the mutation rates to be off. Okay, mutations could be lost. They're saying they will be lost through genetic drift. So you may have some populations that lose mutations just because they, the population changed, got isolated, something like that. And, mutations that were there, they just got lost. And so maybe we're not accounting for that properly. Then another one, and I know this is like getting so boring, so thank you for hanging in, okay? And I do have people that are waiting to jump on and challenge me, so I'm looking forward to that. But I have to get through this because I don't want anyone to think that I'm just going to present all the evidence, in favor of my view and not bring forward what the potential evolutionary explanations could be. Okay, so the next one is oocyte bottleneck. What does that mean? It means that when your reproductive cells are being formed, there's like a bottleneck where the mitochondrial DNA will go down very low and go to make the oocyte. And so maybe we're losing a lot of the mutations there, and we have to account for that. And so that's one of the things that is proposed, even in that paper that I mentioned from 2024 talks about this. And then the other one is that the neutral region isn't as neutral as we thought. So as I mentioned, there's a region, and it's the control region, and in particular hypervariable regions 1 and 2 that they would call the neutral region, that's good for a clock. And there are some things that could cause it not to be as neutral as we think it is. So one that was brought to my attention recently was background selection, which is the idea that maybe it wouldn't be selected against by natural selection. A mutation wouldn't be selected against because it's doing any harm, but it could be because it's close to something else that will be selected against, and therefore it gets selected against too. Okay, so, and there may be other mechanisms that cause a neutral region, the neutral region that we think is neutral, but it could be, not as neutral as we think, over long periods of time due to other mechanisms. So these are the suggested resolutions. And a lot of the papers that I quoted from that talk about the differences between the phylogenetic and pedigree rates, a lot of them do in their studies try to get rid of some of these problems. So for instance, like they will, in order to not accidentally count somatic mutations, the way that they do that is by testing multiple siblings in a family. They have the mother, and then they try to test multiple people in the family. They try to test multiple generations, and then they see if it's like, there's things that they're trying to account for to rule out some of these things. Okay, but and some of the calculations that they do for the pedigree rates do even filter for some of these things. So like they don't even count all of the mutations because they count against them the heteroplasmy and the somatic mutations and things like that. So some of those things are already accounted for in the pedigree studies that I mentioned. But those are the possible explanations for the discrepancy from the evolutionary scenario. And I can't rule these out. Maybe if we consider all of these things, maybe the rates do match up with the phylogenetic rates, with the evolutionary timeline. But there's some reasons to think that I don't fully embrace that idea. And the one reason that's pretty obvious to me is the low mitochondrial DNA diversity in humans. Okay. It's really strange that after all this time, there is such a low diversity. And so the second thing is, this was far off from evolutionary expectations. It was not expected that we would be finding mutations within just a few generations. Not expected at all. So it's far off from the expectations. And now we're trying to come up with explanations for why it's far off. And maybe they're right, but it still is a point against the evolutionary timeline that what we found did not match up. Our observations did not match up with the story. Now, the story it just so happens to match up with is the biblical genealogy timeline. Really interesting. Did not even have to fall on that date that the Bible has for Eve, but it does. So that just seems, really odd to me. And we have the same thing actually with the Y chromosome, but that'll be for a different day. I'm going to bring on Dr. Thompson, who is here to challenge me. Hey, Dr. Thompson, how are you?
I'm fine. How are you doing?
Good.
Good. Can you hear me okay?
Yep.
All right. So we're talking about mitochondria and humans. All right. Can I ask a clarifying question?
Sure.
Does any of your analyses take into account Neanderthals?
No.
Interesting.
And that's just because, look, I'm new to this. I do know that there are, interesting studies now that we have Neanderthal DNA. But I haven't even, the studies that I listed, those are the ones that I've read so far.
Okay.
So, right. I'm not prepared to talk about Neanderthals, but if you want to, I'd be happy to.
Sure. But do you consider Neanderthals to be humans?
Human.
Like part of the human lineage?
Absolutely human.
Great.
Yeah.
So that they I suppose in your interpretation, that they were progenitors or sorry, they were descendants of Adam and Eve.
Yes, I think they lived contemporaneously with, you know, all humans.
So these you take as gospel, so to speak. These estimates from pedigree studies showing that if we take these estimates from pedigree studies with all the caveats, ignoring those caveats, that mitochondrial Eve existed something about 6,000 years ago.
Between 6 and 8,000, yeah.
Does that take into account mitochondrial DNA from Neanderthals?
Right now, no, but I know that there are people have done that, but if you think
I can
that
assure
is a
you it doesn't.
problem, if you think that's a
Yeah,
problem, why don't you tell me
sure.
how? Because there's people that know a lot more about this than me that haven't brought
Yeah,
to me any problem with Neanderthal DNA related
sure.
to this.
Happy to go
So,
to it. So let's kind of before moving on to the Neanderthals, because that is where I'd like to take the conversation. I do want to address a few of the things that you talked about. So,
but just
uh,
let me let you
yeah.
know, Dr.
Mhm.
Thompson, there is someone else waiting backstage.
Oh,
So let's
gotcha.
try to like keep it
I'll keep it
to a
short.
certain,
Yeah.
yeah. Okay.
So it's absolutely the case that those pedigree studies are not. They're measuring mutation rate and they're not measuring substitution rate. And that is obviously a very important thing, right? So if you're
But let me interject really quick
okay.
because
Mhm.
most of those studies are done on the control region and specifically
Sure.
hypervariable region 1 and 2, a lot of
Okay.
them, and those are supposed to be neutral regions,
Yeah.
right? So, okay.
Okay. I'm not familiar with all the studies that you presented. I know a little bit of Parsons 1997 that made a big splash when that came out. I don't know, I don't remember the regions that was looked at in that study. But it was absolutely the case that one, we're looking at somatic cells, right? So we're not looking at things that are being passed on to the germline. That's a very important thing, right? Because mitochondrial mutations, as you mentioned, of course, you have mitochondria in every cell. You don't have just one. You have somewhere between 10 and 100 individual mitochondria, and that each mitochondrion has somewhere between 5 and 10 to 20 copies of the mitochondrial genome in each cell. So and also the mutation rate is much higher in the mitochondrial DNA than it is in nuclear DNA. So it's much more prone to getting many mutations just over the course of a lifetime of an individual. So if you take cheek swab or something to measure mitochondria and you want to look at the genetic makeup of the mitochondria, that's going to be different than what you find in the germline that's passed on to the next generation. So that's already one major issue.
Okay, but
So
now what I would like to know is, are you saying this study did not account for somatic mutations?
the
Because
97
it
person study,
yes.
from what I remember, yeah, I think that
Okay.
that's the case.
Because in I can look for it right now. I have the
Sure.
study. Okay.
Gotcha.
But I do believe that in it would be a major oversight, right, to not account for somatic mutations.
I
And
mean,
I
it
believe
depends on the
and
point of the
I've seen
study.
in many of these
Mhm.
studies that they are accounting for somatic mutations. They're actively doing things to make sure that they are not counting somatic mutations while admitting that it could happen. But
Sure.
it's not like Parsons was an idiot. He
Oh,
did
of
like
course. Right.
an amazing study here,
Yeah.
and he surely thought of this. Okay, like
Yes.
he didn't
No.
overlook
Mhm.
this. Right,
Yeah,
you're
I
sitting
agree.
here with
Like,
this idea, but so let's just say I want to say
but
right now, you're not disagreeing with me. What you're doing is you are disagreeing with the published peer-reviewed literature in
sure.
Nature. Right?
I,
So
I'm
you're gonna
disagreeing with
disagree, you're gonna
the
say this
conclusion
is
that you're
wrong.
drawing. No, okay. There's a difference, obviously, if you're looking at a pedigree study, if you're looking at actual mutation rate of between grandfather, grandson, and the father in between, versus the kinds of mutations that end up actually as substitutions that get passed down through many generations over time and get fixed within the population. Now, the Parsons study just does not take that into account because it's a pedigree study that's over a very short timeline and it's looking at somatic cells. It's not able to look at what's passed from generation to generation over a long length of time, which is exactly what you need for a molecular clock. It's not to say that it's not invaluable information. Obviously, it's important information to know Well, what is the mutation rate of a given region of DNA? If we're talking about the hypervariable region 1 or 2, the mitochondrion, or if you're talking about other aspects of nuclear DNA and what the mutation rate is there, like that's important information. But for molecular clock purposes, we need to know what is the substitution rate and what it kind of selection pressure is happening in for a region, because that's going to be the thing that determines like what the overall substitution rate that we can expect over a long length of time, not just one generation or two generations.
Right. In the neutral regions, they believe the substitution rate is equal to the mutation rate.
Mutation rate.
Okay. So so in but I want to make it clear that right now you're actually not challenging me. Okay.
Mhm.
You
Sure.
are challenging not just Parsons, but you're challenging the methodology of all of these studies because there's and they some of them have slightly different methodology, but you're challenging the actual peer-reviewed literature. So you're saying they weren't right. They were incorrect in their methodology.
I don't think that you are properly relaying what the purpose was of Parsons 1997. I'll have to go back and look at it, but I don't think that I think what you're setting up here is for me to be fighting some sort of straw man, and I don't think that that's actually correct. So, but what I would like to do is share with you, since we're talking about mitochondria in humans, I want to share with you a presentation that I have. Let me bring it
Okay,
open
wait a
quickly.
minute. Hold on. Let me
Sure.
read from Parsons' study. I'll read from the abstract so you can see what he was trying to
Sure.
accomplish. The rate and pattern of sequence substitutions in the mitochondrial DNA control region is of central importance to studies of human evolution and to forensic identity
Sure.
testing.
Yeah.
Here we report a direct measurement of the intergenerational substitution rate in the human control region. So, this is directly saying he is not testing the mutation rate, he is testing the substitution rate. Okay. He is doing it for the purpose of human evolution and forensic identity testing. And by the way, he is using this testing in criminal cases. This
Right.
is the rate that he
But
is
is he
the rate,
referring to
excuse me,
substitution rate?
wait, excuse me.
Please go
The
ahead.
rate that he published, okay, is still being used in criminal investigations. So his rate is being used. That's what we're this is being
Sure.
used in a practical sense. So if it's wrong, he should be in jail.
No,
Okay.
Rebecca, I think that you just are totally misunderstanding this. Look,
Okay.
because like the kinds of mutations that one inherits from their parent is not going to be the same kind of substitutions that we expect to see over many, many, many generations over a long length of time. There's a difference. And he's using substitution there in a way that I think is probably well, at least it's a different way that population biologists think of substitution rates and how that actually gets substituted in the genome over time. That if you're talking about a mutation that gets passed from, say, mother to daughter, that's not the same thing as a substitution that becomes fixed in a population after selection has already occurred. Does that make sense?
I don't accept that. What I would
Okay,
like you to do, since it's been a long time since you read the study, is go
sure.
read the study
I'll
and
need to go
then
back and look
come
at it.
back and
Sure.
say
Happy
whether
to.
he's
Yeah,
talking about the substitution rate
sure.
that we need to talk about
Gotcha.
with evolution. Okay.
So can we look at the presentation that I have for you about
Yes. Is
Neanderthals? And
it
I'll
gonna
make it quick
be
so
like
because I know
10
there's someone else
minutes or
waiting.
less? Is it gonna be 10 minutes or less so I don't
Yeah,
have to
we'll keep
keep
it
my
to
other
10
person
minutes.
waiting?
Sure.
Okay.
Yeah, because
Okay.
I only have like 5 slides.
Okay.
And I don't even intend on going through all of them.
I'm trying to add it to stage. Oh, sorry. Okay, here we go. I
That's
have to
your
remove
presentation.
mine.
There you
There
go.
we go.
All right. So this is a phylogeny based on mitochondrial DNA, including information from Neanderthals, right? So I'm going to bring up, let's see, my laser pointer here. Okay, so you can see this. Great. So the uh let's see. Okay, so it looks like I've got two lasers up there. That's okay. The basically this is a phylogeny built upon human mitochondrial DNA. Okay. looking at the hypervariable region for Neanderthals and also Homo sapiens. So Homo sapiens is shown here. This is our overall phylogeny. The interesting thing when you look at mitochondrial DNA for Homo sapiens is that there's basically a single clade for all the non-Africans across the entire world that nests inside Africa, which speaks to the out of Africa hypothesis that there's much more variability that we find in mitochondrial DNA amongst Africans than we do within all other non-Africans across the entire world. But when you then go to compare it to some samples of Neanderthals, as shown in this study here from 2009, it's just completely outside of the clade. So it shows that there's a distinct separation. And this is a nice illustration of this. So this is from one of the studies from Svante Pabo's lab, who got the Nobel Prize for his work on ancient genomes from 2022, I think. So this is basically showing in the hypervariable region a portion, I think it's hypervariable region 1, the differences that we can see within Homo sapiens to Homo sapiens. So he calls it human to human, okay? And the number of differences as you do like a pairwise comparison, essentially. So there are going to be some humans that have virtually no differences or very few differences within this region, right? And this is the number of differences that we can see in modern Homo sapiens. So it's something like 1 or 2 or 3 or 4 or 5 amino acid nucleotide differences that we're going to see, getting up to around 15 or so. And you can see that there's a distribution that we can see when we look at the entirety of Homo sapiens mitochondrial differences within this region. Now, when you do Homo sapiens to Neanderthals, the distribution is not even overlapping. This is just how different Neanderthal mitochondrial DNA is relative to Homo sapiens DNA. So it's entirely non-overlapping, even though you just said that the Neanderthals are a subset of Homo sapiens. This is an indication that it's clearly outside of the Homo sapiens that exist today, and that if you take into account the mitochondrial differences we see in Neanderthals, that it has to be that the last common ancestor between humans and modern humans and Neanderthals would have to have been a long time ago. And in fact, we can have estimates for this. I'll get to that. So this is what you base in these estimates. So this is actually using a real substitution rate for mitochondrial DNA differences. And within humans here, the last common ancestor, mitochondrial Eve or so, is so to speak, is something on the order of between 200,000 and 300,000 years ago. But if you take into account Neanderthal and Denisovan mitochondrial DNA, the split between modern humans and the rest of the Homo group that we can actually get mitochondrial information for had to have been at least 800,000 years ago. So it means that if you're going to then push back the mitochondrial Eve by just using the pedigree information from Parsons from 1997, which of course ignores all these issues of somatic mutations being the things that are actually recorded. So it's an overestimate of the mutation rate, and also doesn't take into account the actual fixation of substitutions over a long length of time. At the very least, it's going to push it back to at least 15,000 years ago, if you take into account the Denisovans, just assuming the same mutation rate.
Okay, see, I don't think this is a problem for my timeline in the way that you
Okay.
think it is. Okay, because first of all, you're going back
Why not?
800,000. You're trying to go back 800,000 years,
Yeah,
which
just
tells
based
me
on
you're
mitochondria.
using you're using some out there phylogenetic calibration, right?
That's not an out
So
there
you're
phylogenetic calibration. This is
okay.
actually a very robust, very typically used standard way of doing this kind of methodology. We're not
What's the
using.
what are you
Mhm.
using? What is the methodology?
It's explained in this study published in Nature in 2022.
I
Uh,
know, but
uh,
I mean, I don't have time to read that right now, so I'm
oh,
asking you.
sure. I think you should though. So I can send you the link to this.
Okay, but what I'm saying
It's an
is you're trying
excellent
to get me
study.
to accept that this
Yeah.
isn't based on, evolutionary calibrations. Okay, but you're not ready to
Right.
tell me how they actually did it.
I, I can tell you that they did. Yes, I mean this is essentially the phylogeny based on
Oh, okay.
the Yes, the actual substitution rate that we get estimated from using a variety of standards. And the thing is, even if you take your assumption of when mitochondrial Eve must have existed around 6,000 to 6,500 years ago, you're only including modern human mitochondrial DNA in that. If you include Denisovan and Neanderthal DNA in that, well, then you have to then push that estimate back at least 3 times past that. So now you're talking about 18,000 years ago. You have to
Nope.
make that, well, how else are you gonna make it
And
fit?
I'm going to tell you how I get it. Okay.
Okay,
Look,
because it doesn't
because
work.
remember, I'm working with a very short time frame. So most of the mutations that have happened that we're observing in humans right now, what happened in the last few thousand years, right? And
Sure.
so Neanderthal, that's a line that died out. Denisovians, that's a line that died out. These were isolated populations. I don't expect them to overlap with the mutations that have, and they've been dead for a long time, right? So they have been dead for most of the history of humanity, right? According to my timeline. Okay.
Mhm.
So they never developed those mutations. So there's no problem with the overlap. No overlap. So they died out thousands of years ago.
Right, that still doesn't make any sense, right? If we're basing this 6,000-year timeline on entirely this population, how do we get a population that died out, what, 4,000 years ago, 3,000 years ago, according to your timeline, that is this far different from the stock population? We're estimating, if you just use this, you're saying that this entire population had a last common ancestor 6,000 years ago, based on your hyper elevated mutation rates. You have to include this population as well. So how did they get so many mutations to be different from this population?
Well, like I said, in my timeline, most of the mutations have happened in the time that they have been gone, right? So,
They're
that
dead. How did they get mutations? I don't understand.
well, they were in a divergent group,
Right. And they were
right?
divergent for
So,
what, 2,000 years
well,
from the stock population?
I don't know the
Okay.
exact time. I don't know the exact timeline.
3,000?
But what I
Okay.
know is that our DNA does overlap. We all have, most people have some Neanderthal DNA, right? So
Yes, nuclear DNA, but not
they
mitochondrial
lived.
DNA.
I know.
Right.
I know. I'm just
Okay.
trying to say
I'm
that,
just
like,
clarifying for the
the
audience.
idea of Neanderthals, being completely separate from modern humans is an idea based on evolutionary assumptions. We have now found that we share DNA with
Yeah,
Neanderthals.
right.
So there's evidence that there's an overlap. Now, I told you I haven't looked into the mitochondrial studies on Neanderthal DNA. So right now I'm taking what you're saying, like you're presenting this evidence, but you're not telling me the methodology that they used to get their, data. So, this would be something that I would have to look at, but
Sure.
thank you for presenting it. And,
Yeah. Yeah,
but
because it obviously blows up the entire notion that uh that humans shared a common ancestor 6,000 years ago.
I don't think it does.
It's wholly
And by the
inconsistent with
way,
that.
just to let you know, I
Mhm.
believe in a little bit longer of a timeline. So I do the 8,000, like I think it was
Okay.
8,000 years
Well,
because I go with the Septuagint dates.
but
So
this
yeah,
has to push it back to at least 18,000 years.
I don't see how.
Okay, this
Do you
is
have any
based
further
this
evidence to
entirely
share? But
phylogeny.
it could
This
be.
phylogeny.
I
No,
would have to look into
Rebecca,
this.
let me explain. This
Okay.
phylogeny is based on pairwise comparisons of DNA in the mitochondria. Of course, you don't have to just stick to mitochondria. I don't know why we have to just stick to mitochondria. We can look at nuclear DNA as well. It obviously lines up exactly the same kind of phylogeny with the same kinds of timelines as well. But even if we just want to stick to mitochondrial DNA, this is it shows that the divergence of the Denisovans and the other Neanderthals had to have been split from the human beings around 3 times, at least 3 times, if not, no, it's 4 times. So we're talking about 200,000 years. If this is 6,000 years ago, this would then have to be 24,000 years ago in order to line up. if you're
I
assuming
don't
the
know
same
what you're
mutation rate
doing,
that
but the
you are
books are
assuming.
cooked. I guarantee the
There's
books
no
are
cooking
cooked.
the cooks here.
I guarantee. Look,
No cooking the books. This
I,
is not the case at all. Look, I can guarantee you that this is actually the substitution rate. You can see like this, the variation that we see within humans is relatively limited, as you said, with modern humans. But if you include the Neanderthals, there's much more variability that we see because it is a distinct group that obviously was quite divergent between humans, Homo sapiens, for a very long time. That's why there's no overlap. But if you're saying this is all from a common ancestor, which I agree with, that then you have to take into account this entire variation here. You're only taking into account this variation. You're not taking into account this variation in your estimates. And when you
okay.
do, this is then the pedigree that you create. And if you're saying that this pedigree is 6,000 years ago, if you're saying this is 6,000 years ago, of course, in our timeline, we say that it's 200,000 years ago. Well, then this has to be 24,000 years ago if you're going to include these other organisms, which you say are humans. And they're all
Okay.
descended from Adam and Eve.
Well, I don't agree with the timeline, but I
Okay.
do agree that they would have had to overlap at some point, right?
But
And so
you take
now,
into account ancient genomes, it totally blows this up. The idea that there's a mitochondrial Eve, that if you have to take into account Neanderthals and Denisovans, and if you do, it blows up the whole thing.
okay.
Okay.
Well, thank you for sharing your
Happy?
evidence, Dr.
Yes,
Thompson. Appreciate you
absolutely.
coming on. I'm going to let Junior on now because he's been waiting since the beginning too.
Very
So,
good.
all right. Nice
Thanks
to
for
see
having me
you.
on.
Thanks.
Adios.
Hey, Junior.
Hey. All right. This is actually quite fun because I was actually going to talk about Neanderthals as well. So this
Oh,
is quite
really?
perfect.
Oh, cool.
Yeah.
Okay, tell me what you know about Neanderthals because I don't have enough knowledge about this topic to know how to respond fully to what the data he was presenting.
So, okay, so let me just hold on a second. Did it, oh, here, the button here. Okay. Share screen. Cool. So now I'm sharing my screen.
Whoops, did I do it? Okay, there it is.
Can you see it?
Yep.
Cool. So this is from this is actually evolutionary article, like ancient DNA and Neanderthals. And I'm just gonna go down here. Sequencing. Okay, so I appreciate Dr. Thompson comments, but he said that all the bits that don't align with the human mitochondria, the Neanderthal don't align with the human at all. But then he said only looking at a specific region. Now let's read the sequencing the complete Neanderthal mitochondrial genome. Later, they just going on the story about how they successfully were able to actually create a full Neanderthal mitochondrial DNA, not just a small region. And then they go on to say this person sequenced the entire genome of 5 more Neanderthals, and then there's an article from 2009 sequenced from 2 individuals from the Neander Valley in Germany, and another in Russia, another from Spain, all over the place, Croatia. So there's actually already studied that they were able to create, the full sequence of these Neanderthals. And then they go on to say further analysis and sampling or more individuals has led researchers to believe that this diversity was more closely related to age than it was population-wide variance. Now, this is important. On average, Neanderthal DNA genomes differ from each other by 20 bases and are only 1/3 as diverged as modern humans today. So if humans are already very low diversity, Neanderthals are 1/3 of that. They're even more similar, obviously suggesting lots of inbreeding between them. The low diversity might signal a small population size. So basically, we have lots of genetic evidence that Neanderthals indeed they used to inbreed a lot because small population spreading across Europe, spreading across Asia, so a lot of inbreeding happened basically, and they are even more similar between themselves than modern humans are between us. Of course, we believe Neanderthals are humans, but like you said, Rebecca, we can explain that because it's a lineage that died out. It doesn't have to be older. It's just a lineage that died out post-flood. That's why now they were able to sequence the whole genome. Let's see what they actually discovered when they compare. There's this paper called A Complete Neanderthal Genome Sequence Determined by High-Frau Sequencing, 2009, I think. Anyway, they took a Neanderthal, just one Neanderthal, I think from Germany, and it's actually the one mentioned here. It's actually one of the 5 mentioned here, and they were able to actually sequence the whole thing, and they compared with modern humans, and here it is, complete. So when you take the whole mitochondria of that Neanderthal and complete mitochondria of us today, you discover that the average pairwise difference, this is average pairwise difference, by the way, distribution of pairwise sequence difference amongst 53 humans, and then between humans and Neanderthal is the red one. Only focus on the red one because that's between humans and Neanderthal. And the average pairwise difference is 200. What, by the way, I need to explain this. Average pairwise difference. That means that on average, this Neanderthal that died, this lineage of Neanderthal that died, has 200 mutations. That not just 200 mutations, 200 mutations. Sorry, 100 mutations that modern humans don't have in 100 specific sites in the mitochondria that modern humans don't have those 100 mutations in those specific 100 sites, if you know what I mean. So it's completely unique to this Neanderthal. And because the APD is 200, that means that on average, modern humans have 100 mutations in specific sites in the mitochondria that this Neanderthal didn't have. And this is really important, by the way, because keep this number in mind that the APD between and by the way, we can extrapolate this Neanderthal guy to almost all of them. Remember what this paper said here? That they were 1/3 as diverse as modern humans. Basically, they were even more similar to each other than modern humans are. That means we can extrapolate this data to almost all Neanderthal and say that they were very, very similar as APD of 200 as well, when you look at the whole mitochondria. So do you see how similar this is? So you're telling me when you look at the whole mitochondria of Neanderthal and modern humans, just 200? It's basically a human. If anything, this should prove empirically, genetically, that it's a human because it's only 200. That means Neanderthals had 100 mutations that we don't have, and we have 100 mutations that they don't have. That's all this means, the average pairwise difference.
Okay, but let me push back on this a little bit because okay, right now there's only 120. Are we talking about the entire mitochondrial DNA? I can't remember what you said at the beginning.
Literally complete.
Yeah. Okay. So like right now, humans only have 120 between us, right? So
no, but what? Yes, no. That's not. That's just how many mutations have happened since Eve. You know what I mean? Only mutation that happened since Eve. And that just means, Rebecca, if anything, that just means that certain haplogroups have mutated much faster. Do you know what I mean? In the same amount of time, Africans, especially Africans, they're the ones that have about 100, 110-ish, Meanwhile, the rest of the world has an average mutation accumulation since Eve of about 45, I would say, based on a paper that Rob Carter did recently that I have here, actually, about 45 mutations since Eve. And however, these are mutations that have happened in specific sites in both Africans and Europeans and the rest of the world. We're talking about 100 mutations in Neanderthal not found in not even Africans, nowhere now, specific to just to Neanderthal. You see what I mean? Specific in those sites.
Yeah, I see what you mean.
Yeah.
Okay, so but what is are you responding
Okay.
to what Dr. Thompson said? Because it sounds like you're actually going along with what, like, and that's fine. I'm not arguing the data, but I'm saying, do
No,
you
no, I
have a
will.
response
Yeah,
to what?
I just needed to. It's important to get this out of the way first before I actually present the problem with trying to put Neanderthals before Eve. That's my
Okay.
main point. That's
I
my main
see.
point.
Okay, cool. Okay, yeah. So you're saying there's 200 differences, and but they're totally
That
human, which we already know from the artifacts and everything that we found with Neanderthals and the DNA that we share with them. They're human. So you can go for it.
exactly, and so now I guess I can show now the problem. Remember, the APD is 200 between modern humans and Neanderthals, because we can extrapolate to basically most of Neanderthals, as that text said. So 200, let's remember that. So that means 100 mutations in specific sites that we have that they don't have, and they have 100 mutations in those sites that we don't have. So that's important to remember. That's why the APD is average of 200. Anyway, now let's see the problem with trying to put the evolutionists. They have three options. They either, this is what they believe, by the way. They believe common ancestor gave birth to Eve, and common ancestor gave birth to other Neanderthals, right? So that's Eve, and that's other Neanderthal, and then Eve gave birth to all humans today, and Neanderthal died out. That's what they believe. Just to let you know. Hold on a second. So the evolutionists have three options. They either place the common ancestor first, but then the common ancestor is going to have his genome, his mitochondria is going to be a specific sequence, isn't it? He's going to have his own sequence, the common ancestor. Let's always remember the final result. The final result is APD of 200 between modern humans and Neanderthals. Let's remember that. They can either say that it's a single lineage, so this common ancestor gave birth to the Neanderthals that died out, and then the Neanderthals that died out gave birth to Eve, and then Eve gave birth to everyone. So this one is Eve, this one is the Neanderthal that died out, and this is the common ancestor. Okay, sure. But it's only APD of 200. That means that from the common ancestor to the Neanderthals that died, 100 mutations needs to happen. And then from the Neanderthals that died to Eve, 100 mutation needs to happen as well. But it needs to be the exact same 100 mutations in the exact same place in order to make the Neanderthal and Eve have zero difference. And then Eve gives rise to an average of 100 mutations in modern populations, and that makes it so that the difference between the modern populations and the common ancestor, right, is going to be 200. It's going to be 200. They can say that, but do you see how that breaks logic? How that 100 mutations needs to happen, then the exact same 100 mutations needs to undo themselves in order to reach Eve, and then Eve gives rise to 100 mutations in order to make the final result of 200. This breaks chemistry totally. Like, I'm not even gonna you know what I mean? This is option number one, and option number one clearly doesn't work. Now, perhaps you didn't understand. I guess you didn't really understand this because it can be a little complicated. So just let me know if you want me
Yeah,
to.
I don't understand it, but I'll go back and watch it again. Give me option number 2.
Okay. Okay. So they stuck with these three options because the final result is only 200 APD between the Neanderthals full sequence and the modern humans. So option number 2, common ancestor first, but it's not a single lineage this time. It actually branches off this time. Option number 2. So option number 2, let's say that the Neanderthals died out. The Neanderthals died out here, and Eve came much later. Eve is right here. Eve is right here. Neanderthals that died out is here. Okay, so now you have same thing basically. You have the sequence here. The common ancestor is going to have that specific sequence, and then it needs to give rise to oh yeah, sorry, and from Eve gives rise to all humans. So okay, but now the Neanderthals here have to accumulate a certain number of, it's going to have to accumulate those number of mutations again, because the final result is 200. So this Neanderthal is going to have those mutations here. It's going to accumulate 100 mutations here, and but see, now this doesn't make sense, because now the mutations, now you need to say the 100 mutations happen in Eve, and you know what I mean, like these mutations are not the same basically as this one here, and then Eve gives rise to 100 mutations in the modern populations. Hold on a second, let me just bring this out here. So yeah, just like, so it just doesn't quite make sense, because you have this original sequence here, and in order to make so that the humans here have 100 mutations, the modern humans have 100 mutations that this Neanderthal didn't have. You would have to accumulate the different mutations along this path. You know what I mean? So it doesn't, again, it just it doesn't quite make sense, however how you try to put it. Or hold on a
I don't
second.
understand why that doesn't make sense. Like I in either one of these scenarios, I guess I'm not understanding the significance of what you're showing me.
Oh, yeah, I know what I mean. I know what I mean, because it can be it can be a little confusing this here, but the key point is because it's such a small amount of mutation differences, it doesn't, it's only 200. So 100 100 that they have, 100 that we have, because it's so low, trying to put Neanderthals before Eve always, it's an impossibility basically. It doesn't like quite make sense. Now I'm going to show you the only one that makes sense. The only one that makes sense. You could say that Eve came first. Even if you assume the Neanderthal, the sequence of the common ancestor is here, and then it gave rise to Eve, but it gave rise to Eve earlier, and then it gave rise to to those Neanderthal that has 100 mutations that we don't have. Well, at least now, and then Eve gives she gives birth to everyone basically. Well, at least now you have those you got those same mutations here, but you forced to say that Eve didn't receive any mutations from the ancestor, because if Eve receives any mutation from the ancestor, the APD is not 200 anymore, is it? It's not 200 anymore. It's going to be 201 or 202, whatever. So, if it's 200 and you want to say that the common ancestor came before Eve, and the difference between modern humans and the Neanderthals is just 100, which makes it 200, then you're forced to say that Eve didn't receive any mutations from the common ancestor, which basically means the common ancestor is just as young as Eve. So, okay, if they want to say that, because you see what I mean?
Okay, I think I get this one. Yeah, I think I get this one, because you're saying like that it would have to be like a genetic reset on after
Yeah.
Eve. Like Eve didn't have any of those like mitochondria. We're talking about mitochondrial Eve, not biblical Eve. But yeah, okay.
Yeah,
So that makes sense.
yeah,
And
so
I'm probably just not understanding the significance of the other things you said. I don't know. Like I have not thought about this at all.
Yeah, yeah. It's just an interesting thing that I started thinking about after I saw the data that it's just 200 APD, and Dr. Thompson was like, "No, nothing matches. It's all different." No, that's just only when you look at a small region. But when you look at the whole thing, you can see that they are basically humans. Not only that, but after that, I can give links to articles showing that there's archaeological evidence that Neanderthal built boats, that they buried their relatives. It just goes on and on, that they all feel human. Um, but yeah, there's problems with each step that they try to put Neanderthals or some kind of fantasy common ancestor before Eve. It just breaks all logic. And conveniently for us, the only one that actually makes sense for them is to say that okay, Eve came, Eve came first, in the branch. Because now I'm gonna tell you something that makes even more sense. Hold on.
Are you going to talk about maybe the lineages that like the lineage that we have is not from Eve, it is from the flood, the women on board the ark?
No. So now look how practical and how it doesn't break any chemistry or logic or anything like that. You have Eve first, and from Eve, you have a lineage of the Neanderthal that died, that accumulated 100 mutations, and you have lineages that didn't die, which is modern humans that has an average of 100, which makes the APD 200. Which one makes more sense? You know what I mean? So yeah, I think it's quite clear here that yeah. But anyway, after you can go back and rewatch why those three completely just break logic and chemistry. It's an impossibility to try to put Neanderthals before Eve. They can do it, but it's fantasy land. That's the point I'm trying to make. But anyway, so I guess you can ask if you want.
Well, thank you so much. I actually think I will need to think about this and look into it before I can, you know, figure out what's going on. So I appreciate you sharing, and I will go back and rewatch what you presented. There is somebody else waiting backstage, so I would like to
Oh,
bring
sorry.
them on. And thank you so much, Junior, for coming on, and thank you for sharing about Neanderthals.
That's okay. Just wanted to quick say, um, yeah, I'll. Sorry, I don't have time to do it now, but on the comments, I can say why natural selection doesn't really apply to mitochondria. I'm not saying natural selection doesn't happen for all the evolutionists. They're saying, oh, the low variety that we see today is because many lineages got removed and so on and so forth. Well, what I'm trying to say is natural selection does happen in the nuclear DNA, not the mitochondria. Why not in the mitochondria? Because even though you have thousands of copies in each cell, only about 3 copies get passed from mother to child. And this is from the expert that did the best pedigree, which looked at like 116,000 transmissions, literally the best pedigree you can get. And the mutation rate is 1 every 14 generation, super fast, and that's why I really like this pedigree study. And they say that just 3 copies get passed. So you have massive genetic drift happening each generation. It's like a random luck. So you can never have mutations actually accumulating in lineages. It's all like randomly fluctuating up and down, which means you can never have it's never gonna lineages are never gonna accumulate lots of bad mutations in order for selection to work, which means natural selection is actually happening in the nuclear DNA, not the mitochondria. If it does happen in the mitochondria, guess what? They also found evidence that if a fatal mutation happened in the mitochondria, that lineage is gonna die only a few generations after, which means selection cannot remove lots of mutations from the mitochondria, which means the low diversity means Eve is young. I know that was a lot. I'm sorry, but that's why natural selection doesn't work on the mitochondria, which means that our clock is correct. Yeah, she's only a few thousand years old. Yes. Anyway, that's it, Rebecca. Thank you.
Awesome. Thank you so much, Junior. See ya. Okay, before I bring on the next person, there was something I saw in the chat, and I'm sorry I haven't been paying good attention to the chat, but I did see something on there when I told Dr. Thompson they're cooking the books. Like, I know it. And what I mean by that, even though I haven't looked at this study that he's talking about, and actually I haven't looked much into this issue of mitochondrial DNA, with Neanderthals. But the reason I say I can guarantee the books are cooked is what I mean is that it is riddled with evolutionary assumptions. So they're doing some kind of calibration. I don't know what it is because he wasn't able to describe where they were getting these numbers, but they're doing some kind of calibration with the evolutionary timeline, meaning they're using some kind of divergence estimates that assume evolution from the start. And that's what I found over and over again. Even Dr. Thompson, when you brought me that study on dogs and you said this is fully a pedigree study, when I went back and I read that, it actually, yes, it's a pedigree study, but they added in phylogenetic calibrations. So for the getting the dates that you were talking about. So even if it's a pedigree study, they may insert evolutionary assumptions into the data. And so that is what I'm saying is probably happening on that study. And I haven't looked at it yet, but thank you for sharing it. And I was a little bit stronger with Dr. Thompson than I probably should have been. So I'm sorry about that, Dr. Thompson. Appreciate you. And I will bring on Dr. Dan. Hey, Dr. Dan.
Here we go. Hi, how are you?
Good. How about you?
Okay, I'm good. Let's see. I'm interested to see if I look like a square of pixels or if I look like a person.
Yeah, you're pixels for me
I look
right
like
now.
a square of pixels. I don't know why that's happening.
Fine.
I just did a speed test like 10 minutes ago. I'm on a really fast connection. StreamYard just hates me. I don't know why. Whatever. Anyway, so I want to pick up with this Neanderthal thing because I think we're overcomplicating it here, and I honestly have no idea all that stuff Junior was drawing. I couldn't follow what he was saying. But in terms of evolutionary assumptions in the Neanderthal stuff, it's super simple. There are no evolutionary assumptions because all we're doing is sequencing the DNA. We have the DNA and we're sequencing it, right? So you sequence the mitochondrial DNA, and in Homo sapiens, right, all living humans, the maximum divergence between any two people, right, the number of mutations that separates the two most different people is about 120, right? And what the young Earth argument is, we can account for all of those mutations using the Parsons pedigree rate. We can account for all of those mutations in 6,000 to 7,000 years, somewhere in that time frame. Is that a fair representation of how we do this?
Yes, according to that study.
Okay. Mm-hmm. So if we all agree, if in the young Earth model, that Neanderthals are descended from Adam and Eve, and in general, the consensus with, answers in Genesis and whoever else, is that Neanderthals are actually a post-flood population. So they're descended from Noah's 3 sons and the 3 wives, right? So Neanderthals share a common ancestor with humans, according to the young Earth model, right? 4 to 4,500 years ago, somewhere in that range, whenever the flood happened, right? Now,
or 5,500. Yeah.
So the reason this is a problem for the young Earth model is that we can sequence the Neanderthal mitochondria, right? And we find that they're way more divergent than any two humans are today. There's no evolutionary assumption in that. We just sequence their DNA and go, wow, this is way more different from that person or that person or that person than any of them are from each other, right? There's just more differences between their mitochondria and any of our mitochondria than there are between any two living people, right? That's the data that we're working with here, that Chris was pointing out, and Junior actually pointed out the exact same thing, right, with his figures. That's the data we're working with. Are you okay with that? Do you accept that those sequences are valid?
Yes, I do.
Okay, so here's the math then for why this invalidates the young Earth model. We don't have to have any evolutionary assumptions. We don't have to go back 800,000 years. It's just we have a certain amount of variation that exists in Homo sapiens today, and if you use the preferred young Earth rate, you get your most recent common ancestor at the earliest time you can, right? At like 6,000 to 7,000 years ago, which is where you need it to be. You can't do 10 or 12 or 15 or 20 or whatever. That's outside the timeline. Now, some young Earth models can go to 10, but like mostly it's in the 6 to 7 range, right? So if you have to double or triple the amount of divergence you have to account for, even using the same rate, you necessarily have to move that common ancestor backwards in time, right? So if you converge at 7,000 years using the Parsons rate and the 120 differences, then now you have to account for 250, 300, 400 differences. Now you're not within the 7,000 year time frame anymore. Now you're back at 15, 20, 25, right? You necessarily have to move that most recent common ancestor backwards. That's it. That's why Neanderthals invalidate the young Earth model. You can do everything the same. You can do all the math the same, which, to be clear, is wrong. But that doesn't matter because the additional divergence you need kills the model. Also, I think I'm frozen on YouTube right now, so I don't know if anybody.
Oh, yeah, you're frozen, but that's okay. I can still hear you. And you know what? This might be a good objection. And so let me give credit to Dr. Thompson. He might have brought a good objection. You might be bringing a good objection here. The thing I was like getting really, fighting back on with Dr. Thompson is when he started bringing in 800,000 years and that kind of thing. So, this is something, it might be a good objection, and I would like to look into it. I'm trying not to make promises about this because I have like so many things to look into right now, but it might be a good objection. And it is something I'm curious to look into.
Yeah, let me put it this way. Everybody accepts that the Neanderthal sequences are valid, right? Except for one person, and that person is Nathaniel Jeanson, and he's the person who has done this math for the Young Earth Ministries. He's at AIG, but everyone else basically just does his math. Everyone, cribs his work. And his response to Neanderthals is to ignore them. He says they're not valid. His reasons for that are wrong, and it doesn't matter. It's beyond what's relevant for tonight. But he recognizes that if you include the Neanderthals, it does violate the time frame. And his response to that is the Neanderthals are invalid. We don't have to include them. Now, other creationists like Dr. Rob Carter have said, no, the Neanderthal DNA is perfectly valid. We just have to have a mechanism to explain it, right? And other creationists have proposed things like, there's a lot of radiation during the flood, and that accelerated the mutation rates or something. Like there have been, I think, kind of silly proposals to explain that. But in the professional young Earth community, there is a recognition that if you take the calculations that are done at face value, with the Parsons rate, with the Ding rate, with whatever rate you want to use, and extrapolate those backwards, and you include the Neanderthal divergence in there, you're well outside the young Earth timeline.
Okay, I'm going to bring this one to my creation advisor.
Who's your creation advisor? Because this has a thing of like, you've got a bunch of like, kindergarten teachers sitting around going, triangles have 3 sides. You're like, I'm going to bring this to my toddler and see what they say. Like, who's your creation advisor, and why do they get to like do math differently than how it's done in the real world? I want to know who's your creation advisor.
Yeah, I don't know if I want to reveal my creation advisor right now, but if my creation advisor didn't tell me this is a problem, then I would be like, and it is a problem, I'd be very surprised. But
I'd be surprised too if they admitted it was a problem, because they yeah.
Okay, well, it is something I'm gonna follow up on, but I'm actually
with your creation advisor though. Do you understand like why that's a problem, Rebecca? Like, do you understand the epistemic bubble that puts you in? If every time you hear new information, you have to go to, I have to ask the person who agrees with me.
No, I don't take his word for everything. Okay, I fact check him, and I find out that he's usually right. In fact, I can't think of a time that he's been wrong. So it's not about, just taking one person's advice. Actually, I'm a very skeptical person. Even the people I trust most in the world, like my husband, he gets mad because I don't believe what he says automatically. Okay. And I trust him very much. So even the people I trust most in the world, I don't take their word for anything. Okay. I am very skeptical. So, it is something to look into, but I'm actually surprised that you challenged what I said. I mean, you wanted to go down this route of talking about Neanderthals when, you've had an issue with these mutation rate things before with, the pedigree versus phylogenetic studies. So do you have anything that you want to challenge that I said?
Oh, yeah, sure. So first, let's just real quick. This is not like to start a debate about Parsons, just straight up. If you go and read the Parsons methodology, not the abstract, the actual methods in the paper, they didn't weed out somatic mutations. Like just straight up, like if you disagree, go to the methods, find the line where it says we did that. Because they didn't. Just straight up, like that study includes somatic mutations. Just want to clarify that. There's nothing else to say about it. It's just like that study includes somatic mutations in the data, like just to be clear. But the thing that sure, I'd be happy to talk about background selection for a minute, because this is something that is really important here, because what creationists do is they cherry pick the control region, right? Which is only about 10% of the.
Before we go into talking about that, I'm sorry to interrupt you, but before we get off the somatic mutations, because I don't want to forget about that. So you might be right about the Parsons study, and I don't remember off the top of my head if they had a methodology. So I'll take your word for it that they did not weed out the somatic mutations. But do you agree that there are lots of pedigree studies that do take account and try to weed out somatic mutations and still get a fast rate?
Do they try to weed it? You would have to give me a specific study for me to answer that question.
Okay,
I have a folder
could be
full of
wrong
them.
about I could be wrong. I don't remember exactly which one. I know Hayer, I think, did. Santos did. There's several that did actually try to do, and I might be wrong about Hayer, but there's several studies that I read where they specifically discussed that they were taking precautions to weed out somatic mutations by testing multiple siblings and multiple
right,
grandmother, mother, daughter. So
right, but if they test, and this is what Parsons does too, if they test, and they do this in Parsons with the Amish family specifically in figure 2, I think it is, where it's grandmother, daughter, granddaughter, right, in that figure. And what they do is they count the mutations at each step in that pedigree. And they're still, therefore, going to catch somatic mutations when you do that. Because in order to weed out the somatic mutations, you have to do multiple generations. That's the point. If you can do 3 generations, you could do a great-grandparent, you can do 4 generations. If you sample each individual and count all the mutations, you're going to catch somatic mutations. In order to weed them out, you'd have to sequence grandma and granddaughter and not count the mutations that we find in mom. But in Parsons, they explicitly don't do that. They don't do what you would need to do to weed them out.
Okay. I mean, and I'll take your word for it on that. But I mean, are you saying are you saying there are no studies that take this into account?
In a single generation pedigree, it is very difficult to weed out somatic mutations, because in none of these studies, to my knowledge, are you sampling the germline. You're sampling somatic cells, right? Now, right?
Right, but you're sampling multiple generations.
But if you're going from like grandma to daughter to granddaughter, and you're counting all the differences at each stage, that's including the somatic mutations.
Right, but they're going back further than that based on genealogical records and comparing divergent lines.
So
right?
why
So
is it
they're able to do deeper pedigrees because they're going back.
Yeah.
They may not be able to test the blood of that person that they diverged from, but they're able to look back. They're able to look at the sequences of multiple lines with the genealogical records.
Right. And when we do that, and again, this is getting, I want to get to background selection, and that'll be quick because it's pretty simple to explain, I think. But real quick, before we get to that, to be clear, when we do that kind of thing, those are the types of studies that get criticized as having evolutionary assumptions in them when I cite them, because that's like the Sudyal, Tristan da Cunha study, that's the King 2014, Anne of York, Richard III study, right? You just described the exact methodology that I like to cite. And when I cite it and say, look, these are directly known dates with known lineages, and we're sampling from that known genealogical ancestor, and we get a rate that's consistent with a mitochondrial Eve 100,000 years ago, what do people say? They say, oh, evolutionary assumptions. You just described the exact methodology I use, and when you use that methodology, you don't have to go back very far. You go back to the 1800s, you get the slow rate. Yeah.
Okay.
Like what you just described is the correct way to do it.
It wasn't just Sudyal that did that account.
Well, there's a bunch. There's the Norfolk Island one. There's a bunch of studies that do that, and they all get numbers that are consistent with the mitochondrial Eve. The earliest you get is approximately 60,000 years ago, and the latest is approximately 200,000.
Okay, and that would be how many mutations in a generation?
It's approximately one mutation. I'd have to do the math real quick, but it's approximately one mutation every, I want to say 30-ish, I think gets you to about the 60,000, I think it is. I'd have to go back and look. But I think
Parsons
it's about,
was one mutation for 33, and that gets you to the 6,500.
But Parsons was only looking at the control region. He wasn't looking at the whole genome. And because the genome is non-recombining, you have to look at the whole thing. You can't isolate the control region.
Wait a minute. Then why do most of the studies just use the control region?
Because the control region is where most of the mutations are, because it doesn't have the genes. So you can survive the mutations there.
Right. But the problem, and I mean, I'm surprised that you're saying that the whole...
Follow the logic. Keep going.
Well, I'm surprised that you're saying the whole genome, the whole mitochondrial DNA is good for sequencing, because you're always complaining that we're not accounting for selection.
Yeah.
And if we do the whole mitochondrial DNA, then we're going to have a lot of problems figuring out about selection.
You look at the substitution rate of the mitochondria, and the reason you have to do this is it's non-recombining. Now, when you're looking at the nuclear DNA, you can look at individual regions because it recombines. So you can have individual regions that are disconnected to each other because of crossing over, because of recombination. In the mitochondria, it's non-recombining. So this gets us to background selection. This is perfect. This is why this matters, right? So let's look at this. Just think about the mitochondrial DNA as a clock, right? Think about it as a clock face, right? Now, the control region is a really tiny fraction of the mitochondrial DNA. So take from 12 o'clock to 1 o'clock. That's your control region, right? And then from 1 o'clock all the way back around to 12, that's the region where you have all of the genes, and it's super dense. There's almost no intergenic regions in that part, right? So you have a person who has 10 mutations in their control region. And those mutations, they're in the region that doesn't affect the protein, so they don't hurt that person, right? Now, let's just say that person experiences a mutation, like in their embryo, right? When they're during development, they experience a mutation in one of those mitochondrial genes. And that mutation is really bad because it means they can't do cellular respiration correctly. So they die, right? At some point they die and they don't have kids. So that one really bad mutation never gets passed on, right? So we don't see that mutation like over lots of generations, right? That's selection happening. Does that make sense? We're okay with that?
Yeah, totally following.
Okay.
Yeah.
Okay, now when you lose, let's say that mutation happened at 7 o'clock on the clock, right? In that chromosome, it happened down here at 7 o'clock. What happens when that individual dies? What happens to the 10 mutations in the control region? Do those get passed on, or do those also go away?
Well, as you said, this is the background selection. It's linked together. It's gone.
So they go away. So would the rate at which those 10 mutations occurred be equal to the rate at which they achieve fixation?
Oh, okay. I see the point that you're making. Yeah. Okay.
That's background selection. So when you remove the very rare but deleterious mutations, right? They're actually not that rare because there's a lot of germline selection that we never even see. These are actually because it's 90% of the genome. But when we remove those deleterious mutations, you are removing the linked variation because the mitochondrial DNA is non-recombining. So even if those 10 mutations, let's say those 10 mutations, those are probably not the only instance of those mutations in the population, right? There's probably other individuals that have one or more of those 10 mutations. But because somebody with those mutations got a really bad one and that individual died without kids, those 10 mutations are now reduced in frequency in the population, and that increases the time it takes for them to achieve fixation within that population. That causes the mutation rate to diverge from the substitution rate.
Right. But that really doesn't explain that doesn't explain our observational studies of the mutation rates,
It
right?
does, because
obviously we're
they're too
not going to
short.
be testing the people who died out, right? Like if
Exactly,
they
right.
died out,
But
they're
you
not
can
going to
do that, and that's why you use the multi-generation studies. Exactly. In one generation, you're necessarily only testing the people who survive, right? So there's no, you can't detect that effect when you're looking from mother to child, or from grandmother to mother, or from that mother to the next kid, because it's single generation steps, so you can't detect that effect. But if you go back 100 years or 500 years to a known genealogical ancestor, now you can determine, you can feel that effect, right? And that's why, just going back to Norfolk Islands, Tristan da Cunha, Richard III, just within the last few hundred years, right? Richard III is 1400s, it was Anne of York, right? So you're going back 600 years. That's short enough for that background selection to exert an influence. But one generation, you definitionally can't, right? Because if you're only, like you said, you're only surveying the people who are alive. But over longer periods of time, mitochondrial lineages will go extinct.
Okay, right. But if you're looking back, if you're looking at like the lineages that survived, right? And you're going back to where they diverged, like in the Tristan da Cunha, right? Like you're looking at where they diverged. You're the ones that died out are not going to even be part of that, right? So like
Exactly, and that's why the substitution rate is slower, because you have removed some subset of the mutations that occurred. Yeah, exactly.
Okay, well, I did put that as one of the explanations. Like if I don't know if you were watching the whole thing, but I'll pull my slides up again because I put that in my thing as a possibility, right? As one of the possibilities that could help reconcile the discrepancies between the phylogenetic and pedigree rates. So, you know, I recognize that that's one of the explanations given.
Yeah, and that's, again, this is like a direct documentation of it. I mean, the Tristan da Cunha is just one of the studies, but it's very recent, and it's like a 1816. Like, we know the date too, very clearly. So I really like that one. But that's the point, right? Background selection is a real phenomenon. The mitochondrial DNA is non-recombining. Therefore, you're going to reduce the fixation rate of the neutral variation in the control region due to the harmful mutations that occur in the 90% of the genome that is protein coding. That's it. And therefore, that pushes the date of the most recent common ancestor backwards in time. That's a separate problem from the Neanderthal thing we talked about.
Right, but are you... Go ahead.
No, I was just saying it's a totally separate invalidation of the young Earth model.
Okay, are you saying then there is no more discrepancy between the phylogenetic rates and the pedigree rates? Like we've explained it.
Yes, we have explained this, and the papers you're citing, like what was it? Parsons is from what, 97 or whatever. I mean, there's literally a paper in 2009 that draws this curve that says, like, here's what the curve looks like, the fixation rate curve. Here's what it looks like if you account for selection. And it's calibrated which I know creationists think is a dirty word, but calibration is just you have a fixed known thing and you anchor your data on that fixed known thing. So you can calibrate it based on a bunch of fixed known events and then draw your nice curve. And yes, that resolves the discrepancy. It's only a discrepancy if you assume that the mutation rate should equal the substitution rate, but like, that's silly. Nobody thinks
Okay,
that.
so if it was resolved in 2009, then why are there papers still being published in 2024 trying to resolve it?
I don't know, because people are wrong. Like you wave the paper in Chris's face before and we're like, do you disagree with deep? Like, yeah, scientists disagree about stuff all the time. And like most people don't have time to read most papers. So people are doing, mitochondrial diversity studies and they're like, oh, look at all this diversity in this population or whatever it is that they're show, look at this fast mutation rate in this pedigree. And they throw in parts of it. Oh, if you wind this clock backwards, you get 6,000 years. Okay, population geneticists don't do that because because you have to account for other factors, right? You can't just use a single generation rate.
Right, but I mean, are you saying these population geneticists are so incompetent that in 2015, 2024, they're still doing studies to try to reconcile the phylogenetic and pedigree rates?
Wouldn't surprise me. There's people repeating other people's work all the time. Yeah, that happens all the time. Where people, publish a study, it's like, actually, this was already dealt with, but it's, not all papers are famous. Most papers don't get read by very many people. So, like, yeah, that's a thing that happens. If you have a specific paper you're thinking of where they specifically say, we're trying to reconcile this, that'd be great. It could also be the case that they read the 2009 paper and were like, we think they're doing this the wrong way. Here's the right way to do it.
Right. And let me grab the one I'm thinking about from 2024 and just see, what they're saying about it. So yeah, this is Arna Detour. I'm probably butchering that last name, but this is from 2024. And it says the rate of mitochondrial mutation typically cited in the literature is around 20 times higher than that of the sorry, go
mutation,
on.
Yeah. The mutation rate is higher, yes.
Right. So yeah, and it says based on phylogenetic inference from differences between the sequences with a known divergence time. Pedigree-based estimates are substantially higher than those inferred from phylogenies, both in humans and other species, but are based on a limited number of mitochondrial transmissions and have relatively wide confidence intervals. Go ahead.
Yes, that's a bad thing. Wide confidence intervals are a bad thing.
Oh, yeah, I'm thinking because if you have a wide interval, then you're not very confident.
Right. So what you just read agrees with what I've been saying. We can go through it line by line if you want to read it again, like a couple words at a time.
How does it go with what you're saying?
Read it again. We'll go a little bit by little bit.
All right. The entire thing or just what I said about pedigree based?
Start from the beginning. The 20 times faster bit.
Okay. The rate of mitochondrial DNA mutation typically cited in the literature is around 20 times higher than that of the nuclear genome based on phylogenetic inference from differences between sequences with a known divergence time.
Yes, hold that right there. So mutation rate, not substitution rate. Two different things. The mutation rate is very fast. The substitution rate is not. The mitochondria has a faster mutation rate than the nuclear DNA. Yes.
Okay. Pedigree-based estimates are substantially higher than those inferred from phylogenies.
Yes, because pedigrees don't account for selection and use somatic mutations. Yes, that is true.
Okay. So you're saying,
so what was the next?
You're saying this person is quoting this, and
wait, was there
they're
one more
not
line
accounting for any of that. They're like,
wait. Maybe that person was using the words differently from how I understand those words, but the words that the person said right there, totally consistent with what I've been saying. Yes. Actually, read the next sentence, the part with the wide confidence intervals or whatever.
Okay.
Do the next sentence.
Both in humans and other species,
Mm-hmm.
but are based on a limited number of mitochondrial DNA transmissions.
Boom. Pause right there. So
Yeah.
that's talking about pedigree studies, saying we're only looking at a
Absolutely.
limited number of transmissions, not
Yes.
over longer periods. Good.
Yes. And have relatively wide confidence intervals.
In other words, we don't have high confidence in those numbers. Yes. So when you use limited data sets and you're looking at the mitochondrial mutation rate, right, in pedigrees, you get a higher rate of mutation with a very wide confidence interval. Yes, those things are
Okay.
all true. None of that supports the notion that you can use the mitochondria, the mutation rate, as a substitution rate. Do you see that that is the case?
It doesn't say anything about that yet. Okay, but this is setting up the problem so that the study can resolve some of this, right? And so this study is saying, look, it's based on a limited number of mitochondrial DNA transmissions.
Mhm.
And then this study is based on 116,000 mother-child transmissions.
Okay,
Right.
but that's still just
it's
one
basically
generation.
saying, but now we're doing this study that's extensive. It's not a limited number. It's extensive and it's deep.
Read the next paragraph.
Various explanations have been proposed to account for the difference between phylogenetic and pedigree-based mutation rates.
Yeah.
So this is 2024. There's still a difference.
Yes, that's what. Do you understand? Like, is this in the introduction or the abstract?
This is in the introduction.
This is it, exactly. So they're doing the lit review part. They're saying, here's a thing that people have seen, right? This discrepancy. There have been a bunch of explanations proposed to explain it. That's the part of the paper that we're doing here, right?
Yes.
So,
yes.
so what do they say about pedigree mutation rates in that paragraph?
Okay. One is their, this is their explanation. One is recurrent mitochondrial DNA mutations at highly mutable sites that are detected in pedigrees but missing in phylogenies, where each branch typically spans hundreds or thousands of generations.
Okay. Yeah. So you have, so that's saturation, right? Where you have multiple hits at the same site, which I don't think that's that big a deal, but sure. I mean, given the size of the control region, sure, could be, maybe.
Okay. Yeah, and if we keep going with this, they are setting up a lot of the problems that they are going to attempt to resolve in
which
the
is
study,
which is my point, and I'm glad you pointed that out because this is my point. Why quote from that part of the paper as though it's like the important part if they're just setting up the problems they're going to solve?
Well, because the whole point of me quoting from this is to say that this problem hasn't been resolved. This is still an ongoing thing in 2024. You were kind of making it sound like, oh, we figured this out back in 2009.
I mean, we figured it out before then.
We figured this back out in 2009. Why are we still trying to figure it out in 2024?
Because scientists disagree and they're constantly refining like our understanding. I don't know why that's like, there's never just like one paper on a topic and then it's done. Like, that just isn't how it works. I don't know why that's surprising. And also, you're always going to, and as a matter of just like writing in science, you got to sell it, right? So you're always going to write your paper to be like, this is a problem. Here's how I solved it. Even if it's actually a problem that there's already an answer to.
Okay.
Like, that's just how papers are written. And what you ought to be doing is quoting, and this is just not you, this is creationist, like in general. Instead of just quoting the abstract, the intro, if you're going to pick a quote, go to the discussion at the end where they're summarizing their findings in the context of what we know. That's the part.
Well, let's do that.
Where we're going to see their conclusions, right? Do you have a link to this that I can pull it up? Because I didn't catch the author's name when you said it a little while ago.
Yeah. I mean, I really hate trying to search for stuff because I only have one screen. So, but I'll try to get it.
I can pull this up and I can like explain it if I can pull it up. I probably have the paper.
Okay, look it up under Helgeson 2020.
Oh, it's Helgeson. I have that paper. Yeah, it's Helgeson. Okay. Yep, I got that one. Where's Helgeson? I'm spelling it wrong. There it is. Wait, this is 20. I get Helgeson 2015. Is that the one
Okay,
you're talking about?
that's an older Helgeson one. Helgeson is just one of the names on this one. So I
Oh.
mentioned the first name on the paper, which is like Ardatur, Arna.
All right, whatever. It doesn't matter. Anyway,
Okay.
it's what you're quoting at me is the here's the problem we're going to solve, not the here's the solution part of the paper. And if we're going to do
Yes.
this exercise honestly, you need to look at the solutions, not the statement of the problem, right? Now, I
Yes.
know what you're
No,
doing is to
but-
point out that the earlier paper didn't solve the problem. I get that. But engage with that paper on the merits, is what I'm saying.
Right. And I have engaged with this paper on the merits. I only picked
The
that
09 one?
word. I only picked the what?
The paper I referenced, the 09, the correcting for purifying selection paper?
Yes, I did. Look, I don't remember all the details of all these papers off the top of my head, but I did read all of these papers that I'm mentioning. I have read them and paid attention to
Okay.
what they were doing. Okay. So I don't remember the exact thing. I have the 2009 paper right here if we want to talk about it.
Oh, I found the paper. Here you go. Thanks, Chris.
Okay. So let me get that 2009 paper.
Well, look, the whole point is that background, like, we don't have to go through these papers line by line. Background selection is a real phenomenon that exists. And when I explained it a few minutes ago, you're like, oh, okay, I could see how that could work. That's it. That's all there is to it. If we accept that that mechanism is a real thing, then the mutation rate doesn't equal the substitution rate, even in the control region. And that's the ballgame.
Well, it's the ball game for what? What do you mean by that? Like,
It invalidates the 6,000 to 8,000 year time to most recent common ancestor for the mitochondrial DNA.
well, it's a possible explanation, right? But, you know, obviously people are still talking about this. So
We agree.
it's not
Okay,
settled.
let's
It's
go
not
through it
settled.
little bit by little bit. We agree that the most of the mitochondrial DNA experiences very strong purifying selection. We agree on that.
Yes.
We agree that because it's essentially a bacterial chromosome, it's non-recombining.
Yes.
And we agree that if you select out a mutation in the coding region, you also lose whatever mutations are present in the control region.
Yes. Yes. Mhm.
Okay, that's background selection. That's it.
Okay.
Then we agree, and the necessary mathematical consequences of that is that the mutation rate and the substitution rate don't equal each other. That's it. That's the whole argument.
Okay. Is that enough, though? Is
Yes.
that enough to bridge the gap between 600 one mutation per 600 generations and
Why-
one mutation per 25 to 40 generations?
Why are we doing this in terms of mutations per generation rather than time to most recent common ancestor? Because mutations is just they occur, right? A mutation is when the change happens, right? The time to most recent common ancestor is calculated based on the divergence. How long
Okay.
would it take to accumulate the differences?
Okay, so does just the effect of neutral, can you, from your assessment of background selection, you can definitively say that the gap goes from common ancestor 6,500 years ago to common ancestor 6 million
60
years
to
ago?
200,000, yes. Definitively 100% yes. And we can do that without any
Okay, so
evolutionary
you're not
assumptions
doing
at all.
chimp, you're not doing it.
No,
So you're
you
not
can do this
doing
based
human chimp divergence.
Nope, you can do this based on Anne of York. I forget the relationship to Richard III's sister or whoever that they used as the anchoring point for that study. And that's from the 1400s, right? We agree on when Anne of York lived, right? So you don't have to have any evolutionary assumptions in order to make this math work. You just look at the data with known times within the last few hundred years, and you get the slow rate or the phylogenetic rate, whatever you want to call it.
So are you saying that rate based on, whoever of York is like in the 1400s can take you back to common ancestor of human and chimp at 6 million years?
If you were to use that substitution rate that you calculate using Anne of York going to the two lineages that we've determined in the present, the two matrilineal lineages from Anne of York in the 1400s, if you take that rate, you get the human mitochondrial most recent common ancestor, I forget the exact number for that one, but it's outside of 60,000 years ago, right? Whatever it is. Now, I don't think that study applied that rate outside of Homo sapiens. So I can't say what the number would be if you applied it to human chimp common ancestry. But I want to be clear, if they had done that, or if I was to do that, that is not an evolutionary assumption because the causation is going the other way, right? The inference is going backwards, not forwards.
Okay. But the reason I'm bringing up this human chimp ancestry thing is because so many of these papers talk about how they calculated the rate from the human chimp common ancestor split.
And my point is you don't have to do that. You can ignore that. That's perfectly robust methodology, perfectly valid calculations. We can ignore that. We can do this within documented, recorded human history and get a date for mitochondrial Eve that's at least 60,000 years ago.
Okay. And what are the studies that show that?
So possibly the most perfect combination of author and study in the history of science is that the Richard III study, the first author's name is King. It's King et al. 2014. I think it's like Identification of the Remains of Richard III.
Okay. I will look into that.
There's the Norfolk Island study. There's the Tristan da Cunha one, which is Sudyal, whatever year that was. None of these are based on any evolutionary assumptions because we're talking about documented, recorded events within, human history, within the last couple hundred to couple thousand years, I think, in some cases. In the 2009 paper, they used the Canary Islands and Vanuatu, which were settled, I want to say, 2,000 and 3,000 years ago, right? Again,
okay, wait, hold up. First of all, Sudyal didn't. All that paper determined is that the mutation rate would be more than 1 in 36 generations. So I don't think that gets you back to the timeline.
Oh, it does. Because, well, let's look. It's because you can directly look at the variation. I'm just going to do some real quick math here. Let's pick 1 in 30 generations, right? Because I know RawMat likes that number. So 120 differences times 30 generations per difference, right? Times 20 years per generation. And then let's get that and then divide it in half because you get mutations along both lineages. When you do that math, which is how RawMat does it, which, to be clear, is wrong, but when you do it RawMat's way of doing it, you get 36,000 years.
Okay.
Which is 6 times the young Earth timeline.
Now, that's a little strange for me since Parsons said the most recent common ancestor would be 6,500 years based on one mutation in 33 generations. Because
he was only using the control region. But again, it's non-recombining, so you have to use the whole mitochondrial genome because it's non-recombining.
Wait a minute. Hold up. And this is, I sort of pushed back on this earlier, and you gave your explanation, but
how can
it's not a good explanation.
Well,
it's not a good explanation. No, wait, let me respond to this and tell you why.
Yeah, please, because it's non-recombining. So what's the mechanism to count them separately?
Okay, the point is that what most of these studies are doing is testing within the control region. So if you're paying attention to the whole one, then you have to deal with a lot of other factors. But if you narrow in on the control region, which it's not my idea, it's all of these geneticists that are doing this work. They're choosing
I can explain why they do that if you want. I can explain why they do that.
Okay.
Because it mutates, you have more mutations in the control region. Therefore, when you're looking at short-term things like a pedigree, you get more variation and you can see more fine-tuned differences. Whereas if you're using the coding region, there's much less variation there. There's between 50 and 80% of the mitochondrial DNA is identical across all humans. So comparing across those when you're looking at like familial relationships is pointless. There's no differences within the family, right? So you're only looking, so these studies, sequencing is expensive, PCR is expensive, like hone in on the part that's going to be useful. This is the same reason that when you're doing phylogenetics, you have to use different regions of the genome based on the things you're comparing. You can't use like ribosomes to compare apes because they're all identical, but you can use ribosomes to compare apes to bacteria, right? Within a family, just within a family, there's going to be so little variation within like the coding region that it's pointless to use it. But across longer time frames, you can build up the mutations there. Very few of them, but they're still there.
Okay, but that's not the only reason that they're using that region. They're using it because it is believed to be neutral, so that it's not under strong selection, and so it can get a more accurate rate. That's why they're using it.
Well,
so you're saying, no, we should go back to using this one that has all these variables, and
yes, because,
that's
like,
not,
yes,
that's
like,
not
why
what
would
most
you...
of these studies are doing.
Well, because most of the studies aren't, that's, so two things here. One, if we go back a few minutes, we agreed on how background selection works. And like, there's only three things you need, right? There's selection in the one region, mutations in the other region, and linkage between them. Like, that's background selection. We agree that that's a real thing, right? So now the question isn't, does that exist? The question is, how does that affect the rate? Most of these studies, they're not calculating the time to most recent common ancestor. They're saying, hey, look at this mutation rate. If you were to extrapolate this backwards, here's the time to most recent common ancestor. But no one does it that way because there's selection happening. Not in one generation.
Okay, but here's the problem with that and with what were our empirical observations.
No, hang on. No, because the things that I'm bringing up are also empirical observations. The Anne of York, Richard III study, that is an empirical observation. Tristan da Cunha, that's an empirical observation. So you can't say, oh, we creationists use the empirical studies and you use the inferred studies. No. These are all directly observed studies that we're talking about.
Okay, but the Tristan da Cunha one only gets you, the possibility that it's 3 mutations less than Parsons found, right? So it doesn't take you to your timeline.
Yes, it does.
I don't know about all the math that
it absolutely does.
Okay? But you're saying, so 1 in 33 mutations would take you to 6,500 years as the most recent
only for the control. Only for the control.
Okay. But
can you explain to me why over long periods of time, which is anything outside of like 2 or 3 generations, what I'm saying is long, I don't mean millions, I mean anything outside of 2 or 3 generations, can you explain mechanistically why we can exclude 90% of the mitochondrial DNA from consideration, considering that it is non-recombining? Now, in a nuclear chromosome, you pick a region and go because it recombines, all good. You can't do that in the mitochondria, right? It's non-recombining. So on what basis can we say we're only going to take the control region and we're going to extrapolate that backwards and we're going to ignore the other 90%?
Because of your favorite concept, purifying selection. So we already know, right?
But we agree that it affects that.
Coding regions, if you get a mutation there, you could die or be harmed. So you are not going to be reproducing and passing on. Now, you're saying this linkage, this background selection so that'll take away mutations.
I didn't say that. We agreed on that. We agreed that that's the case. To be clear.
I agreed that might be an explanation, right?
Well, okay, let's go through it. Let's just check it again.
But hang on.
No, I want to see if we agree on it, because 20 minutes ago we did, right? So there's the coding region is highly constrained, right? And it's linked to.
You don't need to re-explain it.
We agreed. You're saying like now you're skeptical of it though. Like we agreed that this is a thing that happens.
I have tried 3 times to tell you why it goes against, like why it doesn't work for our empirical observations.
But it does.
Mutations. I'm trying to tell you, but you're not willing to listen. Look, just let me say what I'm going to say. You can argue with it after I say it, okay? But the reason that background selection probably does not make a difference in the studies that we're looking at, okay, is because we are observing the ones who survived with the mutations. They didn't die out.
Thank you.
So that linkage didn't go together. So
no, it did. It's just no, Rebecca. It's not that they somehow magically became unlinked. It's that in the one generation, like you said before, you're only seeing the kids who survived.
And
So you're
that's my
not
point.
seeing
I'm not
you're
saying they
right
became
that we're
unlinked.
not
I'm
seeing the
saying
effects of selection. It's because it's a short pedigree. That's the point. When you expand the pedigree back 3, 5, 10 generations, a couple hundred years, now you see the effects of selection, and that's why you get the slower rate. You explained this perfectly, like a little while ago.
actually that seems like we would have a higher mutation rate. It seems like it would work in the opposite direction of what you're wanting it to work in, because like, let's say these mutations are linked with deleterious mutations, and therefore they're getting selected against. That means we're counting a slower mutation rate than what we should be counting. That means we've lost mutations that we should be counting, so the mutation rate should be higher.
No, because we don't use mutation rate to calculate time to most recent common ancestor. We use substitution rate.
I know, but the whole argument is whether or not this is like, we're talking about why are we using these different regions? In the control region, in these hypervariable regions, it's supposed to be neutral. They believe it is neutral, right? So now
In a single generation,
they're saying
yes.
possibly it's not as neutral. Possibly it's not as neutral as we thought.
Mutations in that region are generally neutral, which is why we see more variation in there. The problem is not selection on the control region. The problem is background selection, because it's linked to the rest of the mitochondrial DNA, which, remember, is 90% of the mitochondria. So assuming, which is a valid assumption, that mutations are approximately equally likely to occur anywhere in the mitochondrial DNA, approximately 9 out of 10 mutations are going to occur outside of the control region. We don't see all that variation outside of the control region because you have strong purifying selection there. Because those two regions are linked, because the DNA is non-recombining, you also lose variation, like we agreed on, from the control region. And that slows down the rate of accumulation.
So that's why you like the whole thing, because it slows it down to where you need it to be.
No,
No.
No. No, you are putting the cart before the horse, and you're assuming that I'm trying to support a position and backfilling rationale to do it. What I'm actually doing is taking into account the factors you need to take into account. You've said repeatedly that when you use the whole genome, you need to take into account all of these other factors. Yes, because those factors are operating over the time periods that we're both concerned with, which is within the last 10,000 years, right?
Which is why most of these studies don't like to do the entire mitochondrial DNA, because there's too many variations, too many like factors that you have to account for. So why are you not okay with using the one that's most controlled, most neutral, the one that we don't have to have all that,
Because
I mean, and that's the other thing, is that we actually know that we've observed that these tick at different rates, basically. So we're trying to pick the one that's the most neutral. So why aren't you okay with using that?
I am not okay with cherry picking part of a DNA molecule that is linked to the rest of the DNA molecule. Rather than ask me, why should we use the whole mitochondrial DNA, creationists need to justify basing their rate on 10% of the mitochondrial DNA instead of the whole thing.
Well, I think I did just justify it, but it's not creationists. It's the geneticists that are doing these studies. And they're saying specifically why they use that region.
Yeah, because they're looking at variation within a family. Yeah.
So are you saying, like, you're not arguing with me, right? You're arguing with these scientists. I am arguing
with the interpretation of their data that mitochondrial Eve existed within the last 10,000 years.
Okay, and I mean, actually, I'm not saying they think mitochondrial Eve lived that shortly ago. I admit,
and they explicitly say their data doesn't support that.
Assumptions.
And to be clear, that the authors in question here explicitly say that their data does not support a recent mitochondrial Eve. Like, to be clear, they are saying that creationists misinterpret our data. Some of them say that explicitly.
I'm just reading the words that they said with no interpretation. I'm saying, look, these are the words they said.
You're reading some of the words.
which
You're reading
I'm
some
doing
of the
what?
words.
I read the parts where they say clearly we can't accept this, right? Like they said this. Parson said,
Did you read the part
this
with the
would-
reasons why?
because it doesn't match up with the
Nope.
evolutionary timeline.
Nope. That's not why. No.
Okay.
That's just dishonest to say that. They don't know. That's wrong. They say you read one of the excerpts before, and it said in the thing you read why this is not valid. And they said this is contradictory with these observations, with these
Yes.
observations. They didn't say, oh, we have to make sure it goes back 100,000 years. We said we have all this other data that points to this timeline. This singular calculation, if you were to do it, would contradict that. Is it likely that the one is wrong or all the rest is wrong? Right? That's what they're saying.
Right.
They're not doing what you're saying they're doing. That's dishonest.
It's my shortening of it to say it doesn't match with the evolutionary timeline. But that's the gist of what they're saying. Okay. And okay, if the evolutionary timeline is right, then okay, they're right to say that. And so I am challenging that the evolutionary timeline, I'm saying that's not what we should be following, even though that's what the authors of the paper still believe in.
The data that I talked about, ignore the evolutionary timeline. Doesn't matter. We all agree on these dates. And we agree on background selection. And you did a really good job earlier explaining why those deeper pedigrees get you the older dates. You did a really good job explaining it, like, I don't know, 30, 40 minutes ago. Like, yeah. And like, those are the methods. Yes. And when you do that, and you said, like, when you do, like, that's how it should be done. When you do that, what years do you get? And the answer is 60,000 to 200,000. Most of them in the 1 to 150 range.
Okay. Well, I think we've exhausted this topic. So, I mean, I don't have anything else to say about it.
I just want to say, go back and watch this because we agree on background selection. And like, we agree on the mechanism. We went through it twice at least. If that is accurate, which we agree is a mechanism, then you cannot get mitochondrial Eve in the last 10,000 years. It causes those two rates to diverge from each other, mutation, substitution. Like, think on that.
Okay,
in addition to the
Here's what I agreed to. Background selection exists, and it's a possible explanation that has been proposed to reconcile these dates. What I don't accept is that background selection has explained it all. There's nothing further to see here. Okay, because if that's true, then why are we still doing studies on this? Okay, so
the answer is because scientists don't stop.
Okay, well,
keep going.
I mean, but they usually try to find things that still have problems to work on, right? Sure. So that's the part that we disagree on.
And well, our understanding of mitochondrial genealogy is certainly incomplete. No one thinks it's complete. So why wouldn't you keep doing studies on this? These types of studies inform all sorts of other things. Everything from disease treatment and transmission to archaeology, right? And does this match with the histories that we think we know? There's all tons of reasons why you would keep doing this stuff that have nothing to do with any kind of uncertainty about the mitochondrial time to most recent common ancestor.
Okay. Well, I'll let you keep the last word on that. And I will say I'm very happy with this conversation because I don't know, you've had a lot of negative things to say about me lately, so I'm glad we were able to have a friendly conversation.
I'm glad we had this conversation too. This was fun, and I hope everyone watching enjoyed that chat. I've been following the chat. A bunch of you were way off base, and if any of you want to talk to me about it, email me, because I'm happy to go through this with anybody.
All right. Well, thank you everybody for watching, and I am going to shut it down now because we've been going for more than 2 hours. So thank you for everybody who came on. Take care.